Hello students. So I'm back with the new biology class and we were discussing about the chapter molecular basis of inheritance. This is the 7th class of this particular chapter and if you remember we were discussing about the mechanism of replication. In our previous class we have discussed about the enzymes of replication. So let us begin the mechanism of replication. Students, you all know that replication is a very complex multi-step process that requires a number of enzymes, protein factors and ions. This mechanism can be discussed under few steps. The first step of replication is origin of replication. In case of prokaryotes, you know, especially say E. coli, the DNA is present in the form of a single circular DNA. Now, replication actually begins at a particular region of DNA, which has a particular nucleotide sequence. This nucleotide sequence is known as autonomic replication sequence. So the special sequence which initiates the process or from where the DNA replication starts is known as autonomic replication sequence. It is called origin of replication. It is called origin of replication in case of prokaryotes or simply ORI. What we call this? ORI. It is the place where the replication begins. See, this is a double strand of DNA and this is the ORI where replication begins. Now, prokaryotes have a single origin of replication, whereas eukaryotes have multiple origin of replication. Say Drosophila has about 3,500 origin of replication. Now, the DNA of prokaryotes functions as a single replicating unit called the replicon. The entire DNA functions as a single replicating unit or replicon. But the DNA of eukaryotes, they possess a number of replicons or replicating units. Now, the replication is a bidirectional process. It is a bidirectional process. So, it occurs in this direction also, in this direction also. Rarely the replication is unidirectional. This is the first step of DNA replication. The second step is called activation of deoxyribonucleotides. So what happens in activation of ribonucleotides? ribonucleotides. Actually, there are 4 types of ribonucleotides that you know, that is deoxyadenosine monophosphate, deoxyguanosine monophosphate, deoxythymidine monophosphate, and deoxycytosine monophosphate. So, with the help of deoxynucleotides and the energy, the phosphate group and enzyme phosphorylase, the nucleotides are changed into triphosphates. What is that? Say the deoxyadenosine monophosphate. They react with the phosphoric acid, and with the help of the enzyme phosphorylase, they are changed to deoxyadenosine triphosphate. This occurs for the DGMP, DCMP and DTMP. So, this process is called activation of deoxyribonucleotides. The third step of DNA replication is initiation. Initiation of replication. Now, for initiation of replication, see the figure here, you will be able to know that the DNA strands must separate. So, this is a double helix. So, these DNA strands must separate for the replication to begin. So, how this DNA strands separate? The origin of replication is recognized by the origin recognition complex, and it attracts the enzyme helicase. It attracts the enzyme helicase, which unwinds the DNA helix and unzips the 2 strands by breaking the hydrogen bonds. ATP is used in this process. ATP, or the energy currency, is used in this process. Now, the 2 strands function differently on the 2 sides of OD. One functions as the leading strands, the other functions as the lagging strand. Now, the leading strands opens in the direction 3', 5', and the lagging strands open in the direction 5' and 3'. So, the separated strands become stabilized in this condition with the help of SSB proteins. With the help of SSB proteins. So, helicase comes and unwinds the strands. The strands are stabilized by SSB proteins, and the topoisomerase, or which is known as DNA gyrase in prokaryotes, it relieves the tension between the 2 strands developed due to unwinding. So, what has happened in initiation step? The original recognition complex is formed, which attracts the enzyme helicase. It unwinds the 2 strands, and SSB protein stabilizes the 2 unwinded strands. Topoisomerase 2, or DNA gyrase, it relieves the tension formed due to the unwinding. So, this happens in the initiation step. The step 4 is priming. It is called priming. So what is priming? Priming is when the replication fork is exposed. See, the two helices are separated in the frontal portion and they are still winded in the lateral portion, and this forms a Y-shaped structure called the replication fork. At the 3' end of the one strand, that is the leading strand, a small RNA piece is synthesized. With the help of the enzyme DNA dependent RNA polymerase. DNA dependent RNA polymerase, or simply called primase, This catalyzes the formation of the RNA primer, a short nucleotide strand of RNA about 6 to 10 nucleotide long. This is required because DNA polymerase cannot initiate the process of nucleotide binding. So after the priming is done, next comes the step called base pairing. So the fifth step is base pairing. So what is base pairing? Now the two strands that have been separated in the area of replication fork now function as the template. So this is one template and this is another template. The nitrogen bases attract complementary phosphorylated nucleotides. So the nitrogen bases in the template strand, they would attract the complementary phosphorylated nucleotide, that is the DATP, DGTP, DCTP and DTTP formed during the activation of nucleotides, and they will be placed in a complementary manner. Suppose the leading strand has A, then in the newly synthesized strand, the thymine will come and lie opposite to A. And if in the template strand we have G, then C will come and lie opposite to the G. In this way, A would form a bond with T and G would form a bond with C. Now, in this process, two phosphate groups will be released because we need actually DAMP, DGMP, CMP, DTMP. So the two phosphates group will be released. Like for example, I say DATP. This with the help of the enzyme pyrophosphatase will form DAMP, required for the nucleotide addition, and along with it, 2 phosphate groups will be released and energy will be utilized. Now the next step comes where the new strand formation takes place. New strand formation takes place. So what happens here? This requires the enzyme DNA polymerase. 3 in case of prokaryotes and delta in case of eukaryotes. Delta in case of eukaryotes. Along with the source of energy like ATP, GTP, TTP and magnesium ions. The enzyme helps in establishing the phosphodiester linkages between the successive nucleotides. This produces nucleotide chain. The DNA polymerase 3 or the delta adds nucleotide only at the 3' end. So this is the 3' end. Only at the 3' end the nucleotides could be added. And they will lie opposite to that of the template strand. The chain elongation will occur in opposite direction. See here with the red arrow, it is shown the direction of the chain elongation. Thus, the parent DNA strand unzippers further to expose new regions of our chain building. And in this way, a continuous strand is formed on the 3' end of the DNA chain. So, this chain or this strand is called the leading strand. Now, in the opposite one or the opposite strand, the 5' end is free, but DNA polymerase cannot catalyze the addition of nucleotides in the 3' to 5' direction. It will definitely catalyze the formation of strand only in 5' 3' direction. So what happens? Short strands of RNA are formed at multiple locations, and from there short DNA new strands are formed on the template strands. In this way, multiple small fragments are formed in the 5' 3' direction on the 5' 3' strand. As I said, this is because the DNA polymerase cannot catalyze the addition of nucleotides in the 3' 5' direction. So, these short fragments which are formed are known as Okazaki fragments, and this entire strand is known as the lagging strand. Now, after this, these small primers of RNA are removed from here and the DNA are ligated using the enzyme DNA ligase. So, DNA ligase catalyzes the addition of the Okazaki fragments. In this way, a continuous leading strand is formed and a discontinuous lagging strands of Okazaki fragments are formed, and then they are joined by DNA ligase. So, these are the steps of DNA replication. But now comes the last step, which is called proofreading. The last step is called proofreading. See students, DNA replication is a very accurate process. Hardly a wrong nucleotide gets inserted in the DNA replication, but that also is needed to be removed if formed. So the incidence is near about 1 in 1 lakh or 1 in 10,000 in some cases. So DNA polymerase 3 is even unable to differentiate uracil from thymine. So sometimes the mismatches does occur. So mismatched nucleotides are removed and placed by correct nucleotide with the help of mostly the enzyme called DNA polymerase 2 or the beta DNA polymerase. So DNA pol 2 in case of prokaryotes and DNA polymerase beta in case of eukaryotes carry out the proofreading in case of DNA replication. So these were the steps of DNA replication or mechanism of DNA replication.